Welcome, my dear friends. In this regard, CPGET 2023 pre-online coaching. Today's our eminent resource person, Dr.P.Manjula Madam Garu, Assistant Professor, Department of Botany, Rbvrr Women's College, Autonomous. Madam is going to explain about the anatomy, plant anatomy. Yesterday, Madam has clearly explained about the plant tissues, tissue systems also. Clearly explained about the tissues and tissue systems. So, my dear friends, listen carefully. Madam is going to continue yesterday class. Madam, please welcome and start your session. Share your screen, Madam. Please welcome.
Okay, sir. Okay, thank you, sir. So today's topic is shoot apex, organization of shoot apex. Sir, is
Madam,
it visible screen?
Visible, but please click on slideshow if possible.
Okay, sir. Okay, so today's topic is organization of shoot apex. So generally shoot apex, it is the apical part of the stem. So in last class, we discussed about the meristems and permanent tissues, meristematic tissues and permanent tissues. So here in meristematic tissues, generally the meristems are present at the shoot apex and root apex. So here this is also important, mainly theories regarding organization of shoot apex and root apex. So today's topic is organization of shoot apex. So generally shoot apex, it is a apical part of the stem and it contains multipotent stem cells and which produce leaf primordia and also give rise to all the aerial parts of the plant. So from the shoot apex, so it undergoes here only in the shoot apex region only these meristematic cells are present and they undergoes continuous cell division and produce the all primary growth of the plant will takes place to this. So that is leaf primordia and it also gives rise to all the aerial parts of the plant such as leaves, branches, flowers, etc. So it possesses the apical meristem and it continues to grow. So the stem, it is generally the aerial part of the plant and that bears leaves, branches, flowers and fruits. So stem, it is made up of nodes and it is made up of nodes and here the node we can identify the nodes and internodes and where the leaves are born and internode. So generally in the nodal region, so leaves are present. So which is the part between the two nodes, it is called as the internode. So the apical meristem is present on the apical part of the root and shoots. So generally meristems, based on the position, so here the meristems are classified into two types, that is apical meristem, lateral and intercalary. So here the apical meristems, general examples for apical meristem is root apex and shoot apex. So it is a zone of active cell division and it is responsible for the growth in plants and also gives rise to primary tissues. So generally in the shoot apex, it contains the apical meristem. So the stem which bears terminal or axillary buds and axillary buds are generally present in the axils of the leaves and which develop into branches or flowers. So here the shoot apex, it is a tip of the stem, so which contains meristematic shoot apex, it contains meristematic cells and the region of the growth. So it develops into lateral branches, leaves and flowers. From here, from the shoot apex region, So the size, shape, and organization and pattern of the tissue differentiation, etc., all are vary in different plant groups, such as in pteridophytes, it may be varies, and in gymnosperms and in angiosperms. So here the it is varies in different groups of the plants. So some of the characteristics of the shoot apex are, first one is it contains the apical meristem, and below which other tissues are differentiated. So it is present just above the youngest leaf primordia, and it grows continuously and produces the leaf primordia. So when the shoot apex, which forms an inflorescence or flower, so the growth becomes determinate. So it will stop, and the growth also becomes determinate or limited. So when the stem is modified into tendrils or thorns. So the shoot apical meristem, it is a site for flower embryogenesis and primordia of leaves, sepals, petals, stamens, and ovaries, and starts developing. So one by one at the time of interval, it is known as a plastocron. So my main here, the theories are important. So the shoot apical meristem generally it contains 4 types of cells. They are stem cells, daughter stem cells, and founder cells, and that initiate the organ development and the organizing center. So apical meristem, which gives rise to 3 types of primary meristem. So we already discussed about this apical meristem, that is protoderm, procambium, and ground meristem. So from the protoderm, which is differentiates into the epidermis, whereas from the procambium, which gives rise to primary vascular tissues, that is xylem and phloem. That is primary xylem and primary phloem. So next one from the ground meristem, which develops into cortex, pith, medulla, and pericycle. So apical meristem, it is responsible for primary growth in the plants, that is increase in the length or height and organ formation. So mainly here to explain this organization of shoot apex. So here mainly 3 theories are explained regarding this apical theory. So they are first one is the apical cell apical theory, second one is the histogen theory, and third one is the tunica carpus theory. So first one is the apical theory. So this theory was given by Carl Nageli and Hofmeister. So according to this theory, so a single apical cell is responsible for the development of all the aerial plant parts. So this theory, so according to this theory, so from a single cell, so all parts of the plant are developed from this single apical cell. So this theory holds good for some cryptogams and such as the bryophytes and some higher algae and some pteridophytes. So this theory is mainly applicable for bryophytes, algae, and some pteridophytes. So according to apical cell theory, so a single apical cell is responsible for the development of all the aerial parts of the plant. So this theory was given by Carl Nageli and Hofmeister. So next one is the histogen theory. So this theory was proposed by Hanstein, and according to this theory, so the shoot apex does not comprise a single apical cell or a group of meristematic cells, and that are not a single apical, not only contains only here the single apical cell, but it contains group of meristematic cells and that are responsible for the development of the whole plant. So according to this theory, so apical meristem, which consists of 3 distinct zones, so they are called as the histogens. They are dermatogen, peribulum, and pleurum. From the dermatogen, so it is the outermost layer and which gives rise to epidermis. And from the peribulum, so it is present below the dermatogen and it gives rise to the cortex and endodermis. Pleurum, so it is the inner core and that gives rise to the vascular tissues and pith. So according to histogen theory, so it is proposed by Hanstein. So according to him, according to histogen theory, so here according to apical cell, from the single apical cell, so all aerial parts are developed. But here according to histogen theory, so it is all aerial parts of the plant, they are formed by a group of meristematic cells. They are called as the histogens. So here mainly 3 distinct zones of meristematic tissues are differentiated, and they are dermatogen, peribulum, and plurum. So from the dermatogen, outermost layer is formed from outermost layer, that is epidermis is formed from the dermatogen layer, and from the peribulum, that is cortex and endodermis will be developed from the peribulum, and from the plurum, that is inner core, that is vascular tissues and pith are developed from the plurum according to histogen theory. So another theory is tunica carpus theory. So this theory was given by Smith to describe the organization in the shoot apex. So according to this theory, that is apical region of the shoot consists of two zones, that is tunica and carpus. So the tunica, it is the outer enveloping layer, and it is made up of small cells that gives rise to the epidermis and outer cortex. So according to this tunica carpus theory, so here it contains two regions, that is tunica region and carpus region. From the tunica region, that is epidermis and outer cortex is formed from the tunica region, and from the carpus, so it is a mass of cells which forming the inner core. It produces the inner cortex, procambium and pith, that is inner cortex, vascular bundles and pith, they are developed from this corpus. So corpus, it is a mass of cells, they produce the inner core, that is inner cortex, vascular bundles and pith. So in last, I think in last previous year's question, 1 minute. Tunica carpus theory explaining the shoot apex was proposed by Carper, Nageli, Hanstein and Smith. So this is the last year, previous year question. That is tunica carpus theory, which explaining the shoot apex was proposed by. So answer is Smith. Okay, so another question, previous year question, that is companion cells are closely associated with. In last class, we discussed about this companion cells. So generally, they are associated with sieve tube elements. So these are the last year questions from this part. So apical cell theory, that is generally apical cell theory mainly it explains in the algae, bryophytes and pteridophytes. It is applicable for apical cell theory, which is applicable for this algae, bryophytes and pteridophytes, and it is a salts, that is single apical cell, which to be it is present, and which from this single apical cell, so all parts of the plant are produced from this single apical theory according to apical cell theory. Next one is, so here apical cell theory is not applicable to the seed plants. So it is only applicable for algae, bryophytes and pteridophytes. So next one is a histogen theory. So so this theory mainly states that the main plant body, it arises from the mass of meristems, and here they are called as the histogens. So three that is dermatogen, peribulum and plurum. So it is applicable the shoot and root apices. So this is more applicable for both root apex and shoot apex. So this apical cell theory is also applicable for both shoot and root apex. So here the dermatogen, it is the outermost cell layer, outermost single layer of the cell, so which usually divided by the radial walls and gives rise to the epidermis. So another one here. So here generally they undergoes radial divide by radial, but here tangential divisions also seen. Tangential as well as the radial divisions are seen in some members like example ficus leaf. So next one is a pleurum. So it is the massive central core of the apices, so which consists of cells which extended in the longitudinal direction. So the cells of this region divide in all planes and gives rise to central cylinder, that is steel vascular bundles, which consisting of pericycle, primary vascular tissues, and medullary rays and pith. So the pleurum, which remains covered by the dermatogen and the peribulum. So peribulum, it is the intermediate zone. So it is present in between, and which is lying in between the dermatogen and pleurum, and it is composed of isodiametric cells from this region. So primary cortex is formed. So here the innermost layer of this cortex, which is differentiated into endodermis. So here the inner cortex and endodermis are produced from this peribulum. So here Handstein's histogen theory thought, though it is applicable in root apex, so it is still criticized in the case of shoot apex of the angiosperms. So mainly for the two regions. So it is histogen theory. It is also applicable for both root apex and shoot apex. But here, due to sharp distinction between the peribulum and pleurum in the apical meristem is absent, and the origin of different regions of the plant body from the sharply defined histogens cannot be demonstrated. So due to which, so it is not applicable in case of shoot apex. So another one is tunica carpus theory. So this is mainly for the shoot apex. So the third theory of structural development and differentiation is the tunica carpus theory, which is given by Smith, and it is supported by Foster. And according to this theory, it is based on the observations on the angiosperm shoot apices. So it is this tunica carpus theory, it is only applicable for shoot apex and for angiosperm. So according to this theory, here the two tissue zones occur in the apical meristem, that is tunica and carpus. So in the lower vascular plants like pteridophytes and even in some gymnosperm, a sharp distinction between the tunica and carpus is lacking. But in angiosperms, so the demarcation between the two zones is prominent. So tunica, it is the outermost covering zone and composed of one or more peripheral cell layers, which enclosing a central mass of tissue. So the cells of tunica divide mainly anticlinally and bringing about the surface growth. So the tunica cells are smaller than the carpus cells, and each tunica layer develops from a small group of separate initials. So the number of tiles of initials is equal to the number of layers of tunica. So from the outermost layer of the tunica, epidermis develops, but other tissues like cortex, pith, so vascular tissues, etc., and may also be developed from the tunica or from the carpus or from both. So mainly from the tunica, it is epidermis is developed and outer cortex. But sometimes, so here these may be along with this. That is maybe sometimes it may cortex, pith, and vascular bundles may be developed from this according to some observation, some scientist. But it is, of course, depends on the plant species and also the number of tunica layers. So generally from the tunica, that is epidermis and outer cortex is produced. So from the carpus, it is the central core and a mass of cells which is surrounded externally by the tunica layers, and here the two regions are different structure and appearance also due to varying in rates of cell division and methods of growth. So the carpus cells are larger and divide in various planes, so that a mass of irregularly arranged cells is formed and bringing about the increase in the value. So the carpus originates from its own initial located beneath those of tunica. So from the carpus zone, which gives rise to pith and vascular bundles and a part of the cortex, that is inner cortex. From the tunica, that is epidermis in outer cortex is developed from the tunica. From the carpus, inner cortex, vascular bundles and pith are developed from this carpus. So other theories to explain the structural organization of the shoot apex are, that is histogen theory. That is, the concept was proposed by Darwin in 1947. So instead of naming the different layers of the apical meristem, so he named different layers like L1, L2, L3, and L4. So the layers could be recognized by microscopically and also by the assumptions based on the orientation of the cell division. So according histogen layer concept, so he named it as L1, L2, L3, and L4. So another theory is mantle core concept. So this concept was proposed by Popham and Chan in 1950. So the term mantle is used for the dome-shaped outer layer of the apex and core for the inner cell mass, which is covered by the mantle. So in this concept, mantle is used for tunica and the core for the carpus. So another theory. So these three theories are main important, but along with this, so other theories also proposed. So mantle and core concept, which is proposed by Popham and Chan. So according to this, mantle means outer layer, that is tunica, that is outer layer, whereas the core is the inner. So from the mantle, that is epidermis, whereas from the core, inner layers, endodermis, vascular bundles are developed. So Newman classified vegetative shoot apex into three types: monoplex, simplex, and duplex. So monoplex means, so here Newman classified vegetative shoot apex. That is, so in monoplex type, so where there is only one initial dividing periclinally, and this type is found in mainly in vascular cryptogams. So monoplex means only one initial dividing periclinally. So it is seen in vascular cryptogams. Whereas a simplex type, in this type, the apex is composed of several initials in a single cell layer, and these initials divide periclinally as well as the anticlinal divisions. So periclinal divisions mean they are the divisions which are parallel to the surface, they are periclinal, whereas anticlinal divisions, they are perpendicular to the surface. So here both divisions, and this type is found in most gymnosperms. Duplex, another type is duplex type. So in this type, several initials in many layers are present in the apex, and the initials of the surface layer which divide anticlinally, while cells of the inner layer divide in many planes. So this type is found in most angiosperms. So here Newman classified these shoot apex into three types, vegetative shoot apex into three types, that is monoplex type. So here in monoplex type, only one initial divide periclinally. In simplex type, here contains several initials and they undergo both anticlinal and periclinal division. So it is seen in gymnosperm. So another type is duplex type, and it is seen in the angiosperm. So here several initials in many layers are present at the apex region, and they undergoes anticlinal, and also outer layer is anticlinal, while inner layer which divide in many planes. So it is seen in angiosperms. So another theory mainly for root apex, that is Carper and Kappe theory. So this theory was put forward by Shoup in mainly for the shoot apex, that is tunica carpus theory, whereas for the root apex, so this theory is accepted theory. So this theory was put forward by Shoup. So it is similar to the tunica carpus theory of the shoot apex, and it is based on differences in the planes of cell division. So the theory says that cells in the root apex divide in a pattern called as a T divisions. So the outer region of the root apex is the, so here outer region it is called as a Kappe, and the cells of this region divide first horizontally. Next, the lower daughter cells then divide longitudinally, that is at right angle to the plane of the first division. So due to which they show T divisions. So thus the plane of the two divisions form the T in a median longitudinal section of the root. So outer layer, which first is horizontal division, next divisions are perpendicular to the first one. So due to which it appears T shape. So the inner region of the apex is the carpus. So in this region, the T is inverted. That is the second division takes place in the upper daughter cells, and this type of division has been found in the members of Graminaceae and Pyagaceae. So here first outer, that is according to Carper and Kappe theory. So Kappe means outer layer and Carper means inner layer. So from the Kappe, that is epidermis, outer layers are formed from the Kappe, and from the inner layer, that is inner cortex, vascular bundles are produced from this Carper. So here also root epiphysis in angiosperm. So dicards, that is dicard root epiphysis are the following three types. That is common type. So this type is found in majority of the dicotyledonous plants. So in this type, three group of initials are found at the apex. That is outer dermatogen, middle peribulum, and inner pleurum. So the dermatogen forms epidermis and the cap. So generally in the root apex, that is root cap is present. So the main difference between root apex and shoot apex is here the presence of root cap in the root apex, whereas in the stem, no stem cap. So the peribulum forms the cortex and the pleurum gives rise to central cylinder. So another one is a ranunculus type. So the members of Ranunculaceae and Juglandaceae and Salicaceae and Casuarinaceae, Leguminosae, so which exhibit this type of root apex. In this type, so there is a single row of initials which ultimately gives rise to various zones of the root, including the root cap. So some of the cells of the root cap which differentiate into epidermis. So another one is Casuarina type. So this type of root apex which development is evident in the members of the families Proteaceae, Casuarinaceae, and in some members of Leguminosae. So in this type, generally two rows of initials are observed at the apex. So one layer gives rise to stele and other to the cortex and root cap. So epidermis which develops from the outermost layer of the cortex. So in certain members of the families Juglandaceae, Rosaceae, and Lignaceae. So that and Teleaceae. So one of these two rows of initials gives rise to the stele and inner cortex, while the other gives rise to the outer cortex and root cap. So the epidermis arises from the outermost layer of the cortex. So in addition to the discussed types in the dicards, there is a fourth type in the monocotyledonous plant. That is, the first type is found in Geophranthes species, and second type is very rare and found in Allium, Sativum, and Aloe vera and Amaryllis, etc. And the third type is found in Himanthes and Coccineus. So mainly here, the main according to, so here also that is root apices in angiosperms also different type, that is dicard type, that in dicards that is common type, ranunculus type, and Casuarina type. In first one, that is in common type, here three layers, three groups of initials are found in the apex, that is dermatogen, peribulum, and pleurum. In ranunculus type, here there is a single row of initials, so which ultimately gives rise to various zones of the root, which including the root cap. So in Casuarina type, here root apex development, it is in this. So generally two rows of initials are observed at the apex. So from one layer, that is outer cortex and root cap and epidermis is produced, and another initial inner, that is inner cortex, stele are produced. So next one is the differences between the shoot apex and root apex. So in contrast to that of the shoot apex, the apical meristem of the root apex, which generates cells not only towards the axis but also away from it to form the root cap, whereas the apical meristem of the root is subterminal in position, as the root cap remains the terminal. So another difference is root apex forms no branches and no lateral structures like leaves. For that reason, the root apex shows no change in morphology. And root apex which grows uniformly, as it never produces nodes and internodes. And the root branches arise endogenously behind the region of the most active growth. So these are the differences between root apex and shoot apex. That is, in the root apex, root cap is present, hence root apex is subterminal, whereas the root cap is a terminal. So these are the some of the differences between root apex and shoot. That is, in the root, there is no nodes and internodes, no branches, and no lateral structures like leaves. So these are the some of the differences between root and shoot apex, and these are the theories which are important for competitive exams. So I will stop here, and in next class we will continue. Sir.
Hello. Hello. Am I audible? Am I audible? Hello.
Sir, yes, sir.
Okay. Madam, thank you. Thank you very much. So tomorrow we will meet. Okay. Right.
Okay, sir.